Today we're going to talk about genetics and what genetics is. So let's just think about it. How are you the same as your family? Do you have the same nose as someone as your family, or same hair color, and how are you different? Maybe you're shorter than some of your family members or taller. So what I want you to do is think of at least 3 ways that you are either different or the same. And keep thinking about those as we go along through our PowerPoint. Here are some words that you need to know. A gene is a region of DNA that helps determine a characteristic. And we talked about how DNA, of course, is in the nucleus, but it's also packaged on the in the chromosomes. An allele is a different form of a gene. In other words, using an allele for hair color. A locus is the specific location of an allele on a chromosome. Genotype are the alleles that are carried by an individual, and phenotypes are the appearance of a characteristic or trait, like eye color. A characteristic that you'll see is an attribute or a feature, okay? Genetics, they're the study of how traits are inherited traits. And what do you think? What genes do we get and from whom? So this is what this study does. Let's talk more about a gene. The genetic material that controls what we look like, our traits. And if you'll remember, those genes were part of the ladder, okay, in that double helix. Genes are found on chromosomes, and alleles are different forms of those genes. We receive one of each allele from our parents. Look at these different traits. Families have things in common because traits are passed from our parents, like eye color, hair color, skin color, and body shape. Then you have inherited traits. You have curly hair, attached or detached earlobes, freckles, allergies. These are all characteristics that are controlled by a gene made up of DNA. Now we also have genes that decide if our hair is going to be colored curly or straight. This information comes from our parents. There are some acquired traits that are characteristics that we get from the environment, like handedness, left or right hand, dyed hair, languages. We learn that out of from our environment, and our attitude. Here's what he found. He crossed a pure breeding white flower plant with a pure breeding purple flower plant. And what he got from this first generation of plants were all purple flowers. And so then he decided to go to the next generation. So he crossed 2 of the second generation purple flowers, and what he found was that he got 3/4 or 75% pure flowers and 1/4 or 25% white flowers. And what was developed after him was something called a Punnett square to help determine what percentage of a trait would an individual get based on its parents. So we're going to take a look at these and what they are. It's a diagram that's used to predict those outcomes of a particular cross or breeding experiment. It's named after Reginald C. Punnett, who developed this. And it's a diagram that biologists use to determine, like I said, that probability of having that particular genotype. So here's what it looks like. It's a larger square with 4 squares in between, with each square counting for 25%. You can have mother's genes on one side and father's on the other. We'll talk about what those letters stand for. So then you have what's allele pairs. When you have capital letters, 2 together, those are homozygous pairs, okay? Then you have, if you have 2 of the same lowercase letters, these are also homozygous letters. These all represent a trait of some kind, and we'll look at those again a little bit in a minute. When you have a lowercase and an uppercase, those are called heterozygous pairs. And again, you've got one. Hetero meaning more than one. Now we're going to look at something called dominant and recessive. When you have a recessive allele, it's overpowered, okay, by like a dominant gene. Dominant ones are going to have capital letters. Okay, these dominant genes are going to overpower these recessive genes. Dominant is always going to be the capital letter. Recessive is always going to have a lower letter. So let's take a look. Homozygous, meaning one, okay, they're going to have the same genes, okay? They're going to have both dominant or both recessive. So here, these are dominant, both capital letters, and these are recessive, both lowercase letters. So in this case, if B is blue eyes, and this individual has 100% of two capital B's, that individual is going to have blue eyes. If this individual has two lowercase b's for each of the squares, then that's 100% brown eyes. So that individual will definitely have brown eyes. Let's look at heterozygous. Now you have both dominant and recessive. So if you'll notice here in these squares, you have, and we're going to show you how to do this, but in this particular one, even though you have a capital A and a lowercase a, that dominant is going to overpower the recessive. So that particular individual is also going to get brown eyes. So here's an example. Let's look at tongue rolling. So here's an example. Let's look at tongue rolling. Tongue rolling has an allele with capital R. Okay? The capital R is dominant. So let's take a look. Your mom,
it's homozygous dominant for the tongue rolling gene. Her genotype is capital R, capital R.
your dad is heterozygous. In other words, dad's going to have a capital letter and, no, he's going to have two.
Heterozygous. So his actually is going to be not two capital R's, it's going to be a capital R and a lowercase r.
Let's see here. Mom's alleles are going to go on one side and dad's on another.
Here's mom's. Here's dad's. And here's how we do the square.
So take a look here.
And we get two uppercase R's. Notice you have dominant in all of those. The dominant, the uppercase R are tongue rollers.
Take a look at that individual. The genotype, which is the traits, 50%
homozygous dominant, half heterozygous. The phenotype, which is the outward trait, 100% tongue roller. Thanks for watching.